We're going to leave skeletal muscle now, and we're going to take a look at the other two types of muscle that we find in the human body. Remember, there are three varieties of muscle. There's skeletal muscle, there's cardiac muscle, and there's smooth muscle. Remember, when we first talked about skeletal muscle cells, muscle fibers, we said that they're unusual looking, they are quite elongated, they are multinucleated, and they're striped, they're striated. When we use those features and we compare to cardiac muscle and smooth muscle, cardiac muscle shares some features with skeletal muscle. Cardiac muscle is striped or striated just like skeletal muscle is. But when you look at individual cardiac muscle fibers, they are much shorter than the skeletal muscle fibers are. And they have a single nucleus inside of each cell. And that nucleus tends to be centrally located. It tends to be located right in the center of the cell. One of the most interesting anatomical features of cardiac muscle is that it's branched. These cardiac muscle fibers tend to have branches jutting out of them, and I'll show you what I mean by that in just a minute. The other interesting anatomical feature is that the individual muscle fibers are connected to each other with a structure called an intercalated disc. We'll talk about this disc in some detail, but the purpose of the disc, the purpose of connecting each cardiac muscle fiber one to the next, is so the contraction of the heart can be tightly coordinated across time. The heart does not contract all together all at the same time. Individual regions of the heart contract one after the other to produce the pumping effect that the heart needs to perform to move blood through the body. So there are some interesting anatomical and interesting functional differences between cardiac muscle and skeletal muscle. And the same is true of smooth muscle. Smooth muscle fibers are also quite short compared to skeletal muscle fibers. They too have a single centrally located nucleus like the cardiac fibers do. And maybe most importantly, smooth muscle is not striated. Smooth muscle does not have the stripes in it that we see in both skeletal muscle and cardiac muscle. Smooth muscle cells have a unique shape to them. They are what we describe as spindle shaped. So they don't look like a typical round cell at all. They are stretched out, but they are much shorter than what we've seen in skeletal muscle fibers. On this slide, on the upper left, you're looking at a drawing that compares skeletal, smooth, and cardiac muscle cells, or muscle fibers. You're also looking at three images that were taken under the microscope of these three types of muscle. So remember, when we think about skeletal muscle, we think about these very elongated fibers. They are striated, they are multinucleated, very carefully oriented in layers and layers of organization. When you look at a smooth muscle cell, and this is not drawn to scale, this is that spindle shape that I mentioned a minute ago, sort of fattened in the middle and then narrows out on each end. The nucleus is centrally located. There's only one nucleus per fiber. No striations here. And then if you look at the cardiac cells, these are perhaps the strangest looking of all, and that's because of these branches. These cells are branched. They are connected to each other at these light colored lines that you see in the image. Those are the intercalated discs that I mentioned a moment ago. So remember, when we look at skeletal muscle, and that's what you're looking at in this image, even at this magnification, you can see the striations in these fibers. And if I enlarge it, you'll see it even more. There are stripes in here. There are striations in here created by the sarcomeres, created by the overlap of thick and thin filaments in the sarcomeres into dark bands and light bands. That's what you're seeing. And of course, you can also see that each individual muscle fiber here is multinucleated, has several nuclei. Now compare that to this image down here, which is cardiac muscle. In some ways, it looks similar. You can see striations in here. You can see striations in here. But you can also see these very odd, strange-looking branches in areas. In other words, they don't look like these nice, straight, side-by-side linear fibers. There's some connections here that we're not used to seeing. We see these centrally located nuclei, one in each cardiac muscle fiber. That's unique as well. You can't really see the intercalated discs very well, but they're in here. There's one right here. It's a dark purple line. There's another one right here. There's one right here and right here. Those are the intercalated discs. They connect these cardiac muscle fibers one to the next to allow for coordinated contraction. And finally, here's the smooth muscle. Very different looking, right? First of all, you don't see these elongated, linear, large cells. You see these spindle-shaped cells, and they're much smaller. And each cell has an individual centrally located nucleus. No striations. No striations in that tissue. This is a drawing of what we just talked about. I like this drawing because it puts all three muscle types next to each other, so we can compare and contrast the anatomy. Skeletal muscle up at the top, great big muscle fibers. You can't even see the ends of them in this drawing. Multinucleated, striated. In the middle, smooth muscle cells, spindle-shaped. Centrally located nucleus, much shorter than its skeletal muscle fiber neighbor. And down at the bottom, cardiac muscle. Ooh, very strange looking cells. Branches coming off of these cells. And all these cells are connected to each other. They're all tightly connected to each other with intercalated discs that are not clearly visible here. So let's start with cardiac muscle. Cardiac muscle, as the name suggests, is found only in the heart. We give the cells their own unique name. We call them cardiomyocytes. Cardio meaning heart, myo meaning muscle, cyte means cell. Cardiac muscle cells. They are shorter than skeletal muscle fibers. They have a single centrally located nucleus. They are striated, which means they have sarcomeres in them. They have the sarcomere anatomy that we see in skeletal muscle. But these cells are branched, and they are connected one to the other with these intercalated discs. Now, if you look inside an individual cardiac muscle cell, a cardiomyocyte, you're going to find lots of myoglobin, just like you did in skeletal muscle. So this would be a red fiber, as we call it, an oxidative type of fiber. No sarcoplasmic reticulum. These cells are so much smaller than skeletal muscle fibers that there's really no need for a central storage organelle for calcium. Instead, when it's time for contraction, calcium gets pumped in across the sarcolemma. Calcium gets pumped in across the membrane from the outside of the cell. The cell is small enough that that works. Now, what's the function of cardiac muscle? Well, that's pretty clear, right? Cardiac muscle has to provide a coordinated wave-like contraction through the heart muscle that allows the heart to contract in a way that it pumps blood throughout the body. Here's a drawing again of several individual cells in cardiac muscle, side by side with another image taken under a microscope. The two features that really jump out at you are the fact that these cells are branched and that they are all connected to each other with these dark purple lines, these intercalated discs. So in this drawing, you've got a cardiac muscle cell, a cardiomyocyte, starting right here. It stretches down this way. It also stretches this way. These are very unique shaped cells. They are branched cells, and we're not used to seeing that in a cell. They are striated. They are striped, and the reason they're striated is because they're full of sarcomeres, just like we saw in skeletal muscle. Let's take a minute and talk about the structure of these intercalated discs. These discs are actually part of the sarcolemma. I'll show you a drawing of these in just a moment, but for now, understand that they have two key structural features. Each disc that connects two cells, each disc has two parts to it, in other words. It has what we refer to as gap junctions, and it has desmosomes. So a gap junction is a connection point between two adjacent cells. It's a place where the membrane of two cells is brought together in very close connection. Remember, we said that when you bring two membranes together in cells that are capable of conducting electricity, we call that a synapse. And muscle, like nerve, is able to conduct electricity. So this is a particular type of synapse. This is what we call an electrical synapse. The electricity, the action potentials in one cardiomyocyte will be allowed to travel to the other, the next cardiomyocyte, through the gap junctions. These are junctions that have a gap in them. They're structured in a way that creates a tunnel between these two cells. So it allows for electrical current to travel down one cell through the tunnel and quickly get to the next cell without a delay. Because of these intercalated discs, and particularly because of these gap junctions, we describe another type of functional unit of contraction in cardiac muscle. Remember, we said that the sarcomere is the fundamental unit of contraction in skeletal muscle. Well, there's also a functional unit of contraction in cardiac muscle called a syncytium. All a syncytium is a group of cells that function as a unit. So inside the heart, there are groups of cells that work together. They contract together, but allow for that contraction through the whole heart to occur in a wave. So one syncytium contracts, and then the next contracts, and then the next contracts, so that a wave of contraction travels across the heart. So you get this very coordinated pumping motion in the heart. So the gap junctions are present in an intercalated disc, as are desmosomes. Now, we've talked about desmosomes before. Remember, we talked about desmosomes and hemidesmosomes when we talked about the integument. All the desmosome is doing in the intercalated disc is holding adjacent cardiac fibers tightly together. It's anchoring them together. Okay? We need those two cells to be attached. We need cardiac cells to be attached to each other again so that the electrical activity can travel very efficiently through the entirety of the heart. Let's take a look at an intercalated disc. Down at the bottom here, you're just looking at an actual image of cardiac muscle. You can see the individual cells here. There are several of them. You can see the branching that goes on between the cells, and you can see these dark lines. These are intercalated discs. And what these discs do is they connect cells together. So these cells aren't just lying one next to each other. They are physically connected one to the next, end to end, by these structures. So this is an intercalated disc right here. So that means that this right here is the end of one cardiomyocyte, right here. You can see sarcomeres in here. And over here, this is the membrane of another cardiomyocyte. So here's another cell right here. Inside of one cell, inside of the next adjacent cell. Here's the intercalated disc, and it has two parts. It has this region here. These are gap junctions, and this is a desmosome. Remember, the desmosome has the job of just physically anchoring these two membranes to each other, just like we saw in the integument. Down here, the gap junctions are providing a tunnel. And that's what they're showing us. These two membranes are being brought very close together, and a tunnel has been created so that as an action potential travels down this membrane, it's going to just pop in and travel through the gap junction and go right into the next membrane. It's a little purple tunnel. Purple in the diagram, of course. It allows for electrical impulses to be transmitted from one cell to the next, from one membrane to the next, with virtually no delay. And that's critical for the normal function of the heart. Now, the other thing to know about cardiac muscle, to know about the heart as an organ, is that there are specialized cardiomyocytes located in certain regions of the heart that are referred to as pacemaker cells. So there's a region in the right atrium, right up here. This is an image of the heart, and the heart has been cut so that we can peek into the four chambers. We've got a right atrium, a left atrium, a right ventricle, and a left ventricle. These are the cavities, the chambers inside of the human heart. You can see the blood vessels, the great vessels surrounding the heart here. Some are bringing blood to the heart, some are carrying blood away from the heart. There's a region up here in the right atrium that's colored in blue that's called the sinoatrial node, or the SA node. There's another one called the AV node. These are regions where these pacemaker cells live. And what makes a pacemaker cell really fascinating is that they are self-excitable. They have a trait, a characteristic called autorhythmicity. They are capable of reaching depolarization by themselves. They are self-excitable. They bring their own membrane to threshold. They generate their own action potentials. And the pacemaker cells in your heart fire off action potentials at intervals from the moment your heart forms, when you're a fetus, until the moment of your death. So these are amazing, amazing cells, and they're located again in these particular regions of the heart. so that, again, the contraction of the heart can be done in a very coordinated fashion. The heart does not contract all at once. It's a wave of contraction from region to region to region, from syncytium to syncytium to syncytium. Now, even though these cells generate their own action potentials, it's important to know that there is nervous system impact on the heart as well, particularly the autonomic nervous system is in touch with, in communication with the heart. Remember, if you're not familiar with this term, the autonomic nervous system is the involuntary nervous system. This is the nervous activity that you and I don't think about. It happens without our conscious input. So the autonomic nervous system is in tight communication with the heart, and it controls how and when and at what strength the heart contracts. Hormones are also critical, as are various pharmaceutical drugs, in terms of impacting the functioning of the pacemaker cells and the rest of the heart. All right, we're going to leave cardiac muscle and we're going to travel to the smooth muscle to finish up the lecture today. Smooth muscle, like cardiac muscle, is an involuntary type of muscle. Smooth muscle is contracting and relaxing outside of your voluntary control. It's located in several regions of the body. It's found in the walls of hollow organs, things like the bladder, the stomach, the intestines, the uterus. It's found in various passageways, sometimes in and out of those hollow organs, the esophagus, the trachea, all the bronchi down in your lungs, your ureter, your urethra. The blood vessels also have smooth muscle in them. But in addition to these organs, smooth muscle is also found in your eyes. Smooth muscle is responsible for the movement of your iris, controlling the size of your pupil, for the movement of the lens when you focus on objects. It's in the skin. Remember the arrector pili muscle that can raise hairs on your skin. Smooth muscle cells, again, are unique in that they are spindle-shaped. They're much shorter than their skeletal muscle fiber counterparts. They have a single central nucleus. They are non-striated. And they are providing the function of involuntary muscle contraction in these locations throughout the human body. Now, smooth muscle, unlike skeletal muscle, smooth muscle has to be able to provide muscle contraction in these organs for long periods without really a lot of significant rest. So generally speaking, smooth muscle is going to be generating a lower level of force, a lower level of tension for longer periods of time. And just like cardiac muscle, smooth muscle is under the control of the autonomic nervous system, the involuntary nervous system. It's under the control of different hormones and even a lot of local factors, things like stretch, for example. If you think about your stomach, when you put too much food down in your stomach and it stretches out, it's uncomfortable, right? Your stomach will respond. The smooth muscle in your stomach will respond when those stretch receptors are activated, and they send that information to your central nervous system. It's important to note that smooth muscle fibers are literally thousands of times smaller than skeletal muscle fibers. So the way that they contract is going to be very different from the way that skeletal muscle fibers contract. That and the fact that they're not striated, they don't have sarcomeres in them. They have no striations, they have no sarcomeres, but they do have thin filaments and thick filaments. They do have these actin-based thin filaments and myosin-based thick filaments. They're just arranged differently. Smooth muscles also use a type of filament called an intermediate filament. These intermediate filaments are found right underneath the membrane, right underneath the sarcolemma, and they are arranged like a net, like a mesh. The myosin is going to come in contact with the thin filaments just like we saw before, and it's going to slide the thin filaments, causing the thin and thick filaments to shorten, essentially, as they slide past each other. The filaments themselves don't shorten, but the arrangement that they're in gets shorter, just like we saw sarcomeres get shorter in skeletal muscle. Now, I said a moment ago that smooth muscle contraction is regulated very differently than skeletal muscle. There is no troponin and tropomyosin on the thin filaments. Instead, there's a protein called calmodulin. And the other thing to know is that this network, this mesh of intermediate filaments, and again, I'll show you an image in a minute, all of this is anchored to the sarcolemma. It's inside, it's on the inside of the cell, but it's anchored to the underside of the sarcolemma with these structures called dense bodies. Like cardiac muscle, the majority of the calcium that's going to be used to control contraction isn't coming from sarcoplasmic reticulum. There is sarcoplasmic reticulum in smooth muscle, but it's very poorly developed. So it doesn't play nearly as important a role as it does in skeletal muscle. And again, primarily because smooth muscle doesn't need this central supply of calcium. The cells are small enough that they can simply pump calcium in from the outside of the cell. Take a look at the drawing on this slide. This represents a piece of two smooth muscle cells side by side. So the sarcolemma, you have to imagine, is gone. We've taken it off. And what we're seeing here is this mesh, this network of fibers that surrounds the rest of the cell. What you're looking at in green here, this is thick filaments, myosin-based thick filaments, and in orange, these are thin filaments. The pink regions, those are the dense bodies. The black lines, those are the intermediate filaments. And this meshwork, this netting as it looks like, surrounds this the contents of this cell in 360 degrees. It would be located right underneath the plasma membrane, the sarcolemma. Now, during contraction, what happens is the thin filaments are pulled over the thick filaments, just like we see essentially in sarcomeres. So essentially what's happening is all the filaments have overlapped each other. This structure has gotten much shorter. And as that happens, it pulls on those anchoring points, those dense bodies. It pulls on those intermediate filaments, and you get sort of an overall squishing down of that network on top of this cell, and it causes the cell to contract, to shrink down. Very, very different mechanism compared to a skeletal muscle fiber. On this slide, we're looking at flowcharts. We're looking at two of them, smooth muscle on the left skeletal muscle on the right. I like this diagram because it puts side by side the process of contraction in these two types of muscle. Now, it all starts with excitation, right? It all starts with a stimulus from the nervous system, an action potential or multiple action potentials arriving down the membrane of a neuron, down the axon to the branches. And remember, in skeletal muscle, that activity is going to cause a release of acetylcholine at the neuromuscular junction, the generation of action potentials in the sarcolemma that are going to travel down T tubules and trigger the release of calcium from the terminal cisternae of the sarcoplasmic reticulum. What's happening in smooth muscle is different. In smooth muscle, that rise in calcium is coming from the extracellular fluid. Calcium is flowing through channels, ion channels in the sarcolemma, from outside of the cell to inside of the cell. Remember, skeletal muscle is under the action of acetylcholine. Smooth muscle is a little bit different. Smooth muscle can respond to different chemicals than skeletal muscle does. We know that in skeletal muscle, the calcium binds to the TNC of troponin, causes it to change shape and move the tropomyosin out of the way. So the myosin binding sites are exposed. Over here under smooth muscle, you can see, I love this box, series of biochemical events. Some magic happens right here, right? Actually, the magic is not all that hard to understand here. So what the calcium is doing in smooth muscle is binding not to troponin, but to that molecule called calmodulin. And when calcium binds to calmodulin, it creates a shape change. And that new complex of calcium and calmodulin is going to activate a type of enzyme called myosin light chain kinase. Kinase enzymes phosphorylate things. And what's going to get phosphorylated here is the myosin heads. The myosin heads are going to get phosphate groups added onto them, and that's going to cause them to change shape. And it's the shape change that's going to allow the myosin heads to bind to the actin in the thin filaments forming cross bridges. Remember, on the skeletal muscle side, it's calcium binding to troponin that causes a shape change that pushes tropomyosin or pulls tropomyosin out of the way. That uncovers the myosin binding sites. And then you can have the heads binding to the actin in the thin filament, forming cross bridges. In both of these cases, as long as there's ATP around, you got to have ATP for all of this. But once the myosin heads can come in contact with the actin in the thin filaments, you're going to have those power strokes, and you're going to see the thin filaments sliding past the thick filaments, and contraction occurs. Doesn't matter if you're looking at skeletal muscle or smooth muscle. It's just happening slightly differently because in skeletal muscle, the thick and thin filaments are arranged in sarcomeres, and in smooth muscles, you don't get that. You get this mesh. You get this mesh that lies all over the interior of the cell. Here are the dense bodies right here. Those are the places where the meshwork is attached to the sarcolemma. You can't see the thick and thin filaments specifically here, but you can see intermediate filaments drawn in. They're also showing us this structure called the calveoli here. Don't worry about that term. I'll tell you that calveoli are just these little depots where calcium is brought into the cell, particularly where the calcium channels are located. Don't worry about that term. The key thing is that once contraction begins, where the thick filaments are pulling on the thin filaments, sliding the thin filaments, those structures are getting shorter, and they're pulling on the dense bodies. And the dense bodies are pulling on the intermediate filaments, and this happens. The meshwork all around that cell just gets tighter. It's like you're pulling on a drawstring, and you're tightening up this mesh all around that cell. And when that happens, the whole cell contracts. So smooth muscle, skeletal muscle, they're very different anatomically from each other, but they use a lot of the same players: thick filaments, thin filaments, calcium, ATP, and contraction.